Plant pathology preparation model question paper number 15, MCQ based. So I'm NH Shankar Reddy, PhD plant pathology. Knob shaped haustoria or knob like haustoria is produced by albugo, peronospora, erisife and all the above. So if you see here, albugo and peronospora actually comes under Womeister's group of fungi, right? So erisife generally it is a genera of powdery mildew. It will produce chains of conidia. So the answer is actually albugo. Albugo only produces chain knob-like haustoria. If you see, imagine this is it will produce a knob-like haustoria. This kind of haustoria is actually produced by albugo. If we see in Womeicetes like Pythium, Phytophthora. While studying Pythium and Phytophthora, next later one more fungi will come, albugo. Generally, it causes white rust in Amaranthus or Cruciferous, Albugo bliti or Albugo candida we might have studied. So generally knob-like haustoria is produced by albugo. Second one, aplanospores can be observed. So which of the following pathogen that can produce aplanospores or aplanospores? Aplanospores means spores without flagella. So generally if the spore contains flagella, we can call it as a juice spore. So if it contains no flagella, then it is called as aplanospore or aplanospore. So Pythium and Phytophthora obviously produces juice spores which having flagella, and there is another fungi called Rhizophus. In Rhizophus, it will produce a spore which is not having the flagella. So the answer is Rhizophus, where if you see in the life cycle of Pythium and Phytophthora, both the fungi can produce flagellated spore called juice spore. Okay, in Pythium and Phytophthora, both fungi can produce sexual spore as a oospore. The next one is amphigynes type of anthradim is observed by. We know very well anthradim is a male reproductive organ. If it is a female reproductive organ, woogonium. If it is a Womycota group of fungi, it is woogonium. If it is a Ascomycota group of fungi, it is called Ascagonium. So generally anthradim is the male reproductive organ. So this amphigynes type of anthradim is produced by the following fungi like Pythium, Phytophthora, Syncytium, Plasmopara. So generally if we see in the life cycle, so the special feature of Pythium is it will produce globose slug, sorry, it will produce a vesicle. Actually the juice spores are formed inside the vesicle. That is a special feature of Pythium. So Pythium normal kind of anthradim only, that is a paragynous type of anthradim where the male and female reproductive organs are formed by side. So imagine it is a paragynous type where male and female are formed side by side, gametangel, where this fertilization tube is developed from male to female, and this male gametangel is going to female gametangel. That kind of reproduction can be observed in Pythium. If you see Phytophthora, the special feature is amphigynes type of anthridium, which means where the male reproductive organs form a globose structure abound this female reproductive organ, this woogonium form a globose structure above the antheridium is known as amphigynes type of antheridium. So the next one, sexual stage is also called as perfect stage, imperfect stage, teleomorph, both A and C. Here the answer is both A and C, because the perfect stage or teleomorphic stage both are the synonyms of sexual stage. So imperfect stage means asexual stage. Asexual stage is also known as anamorph. Teleomorph means sexual stage. Again, this perfect and teleomorphic stage are the synonyms, are the other names of sexual stage. The next one, cruciferous type of division can be observed in. So Syncytium, Plasmodiopora, Pythium and Aphanomycetes. In the again same. So this kind of couple of questions are exactly related to life cycles, right? If you know the life cycle only, then only we can answer here. So anyway, when it comes to this cruciferous type of divisions, Plasmodiopora life cycle, we can observe the cruciferous type of divisions or continuous divisions. So these cruciferous types of divisions are generally observed in a Plasmodiopora. Syncytium, Pythium, Aphanomyces, there is no such kind of cruciferous divisions or can be observed. So the next one is example of sexual spore. Here ascospore, oospore, ascospore and basidospore all are sexual spores only. For example, if I want to tell you examples of sexual spore, oospore, ascospore, basidospore, zygospore. Here there is a small difference. Oospore and zygospore. Oospore is actually produced by Womycota group of fungi. Zygospore is actually produced by the Zygomycota group of fungi. Both are diploid sexual resting spores. I repeat, diploid, 2n condition, sexual, resting spore. They will take rest, like survive in unfavorable tension during resting, like a resting spore for your understanding. So the next one is ascospores and basidospores are haploid non-resting spores. These are also sexual spores, but haploid non-resting spores. Ascospores are produced by Ascomycota group of fungi, whereas basidospores are produced by Basidiomycota group of fungi. The next one is mycelium arranged into loosely or compact woven tissues is known as plectenchyma, parenchyma, prosenchyma, and rhizomorph. Generally, this plectenchyma only where this loosely or compact woven tissues are arranged. If you see this rhizomorph, this is a combination or a compact arrangement or combination of hyphae or mycelial threads. This is called rhizomorph. For example, if you see the fungi like Armillariella amelia or Ganoderma, such kind of fungi will produce this structure is known as rhizomorph. This is the resting structure. Majorly it is produced by Armillariella amelia. That is a very important one. So this Armillariella amelia we might have studied in mushrooms. So this Armillariella amelia bioluminescence fungi or foxfire fungi where it emits lights. Such kind of things we might have studied in the mushroom section. Actually that also falls under the mycology also. But here when it comes to this question, mycelia arranged into loosely or compact woven tissues, that is example of plectenchyma. A systemic fungicide used for the control of rust disease. Generally here Vitavax, Plantvax, Furadon, Fosatyl aluminum. Generally, I think I have already discussed in the previously published videos, especially in the section of mycology. So generally there are two important diseases when it comes to this Basidiomycota, rust and smut. So rust, generally the general fungicide that can be recommended for the rust is Plantvax, that is oxycarboxyn or Plantvax. Rust means oxycarboxyn. The trade name of oxycarboxyn is Plantvax. If it is a smut, the recommended fungicide is Vitavax. Normally a carboxyn, the trade name is Vitavax. On the right side I have mentioned one picture, so that will help us to understand about the rust diseases. Generally the rust produces spore called iridospore. Iridospore is otherwise called as repeating spore, otherwise called as summer spore, which are actually n+n in condition, which are golden brown in color, generally single celled. For the management of all kinds of generally rust disease, oxycarboxyn can be recommended. So the trade name of oxycarboxyn is Plantvax. So when it comes to the next one, smut generally produces teleospores. Teleospores are the resting spores, otherwise called as winter spores. Again the teleospores are n+n in condition, so black dark brown in color, generally bicelled. So this teleospores are also known as the teletospore. For the management of smut disease, we can recommend a fungicide called carboxyn, that is Vitavax. So rust disease generally occurs localized infection. When it comes to smut, systemic infection where inflorescence or entire the grains is generally infected. This is general understanding about rust and smut disease, so that it will be very easy to answer other questions also. I think this diagram will greatly help to understand about this rust and smut diseases. So since the answer for the question is a systemic fungicide used for the control of rust disease is Plantvax. So rust means Plantvax, smut means Vitavax. So the fungus genus that can cause anthracnose diseases generally Glomerella, Elsinoi, Coltrotricum, all the above. Generally the answer is all the above, because other names are synonyms are teleomorphic stages. If you see Coltrotricum, or we can also see this Glomerella, Elsinoi. So Coltrotricum, all this generally causes anthracnose disease only. For example, Coltrotricum generally causes anthracnose disease in mango. If you see Elsinoi amphelon, which causes anthracnose in grapes. So almost all are the genera of anthracnose only. The answer is all the above. So mature virus particle is known as viroid, prion, virion, adult virus. Generally viroid means so that is completely a divide of protein code, that is called viroid. Prions means infectious protein particles or protein that is the infected protein, this is known as actually prions. So that what can I say, that malfunctioned protein is generally known as this prions. Virions means the immature virus particles is known as virion. So the next one is the coconut kadang disease is transmitted by virus, nematode, viroid, spiroplasma. So this coconut kadang is a viroid disease. So the next one, and this coconut kadan is also the smallest virus that is reported so far, which contains 246 nucleotides. So the next one is groundnut bud necrosis virus is transmitted by this groundnut bud necrosis virus is transmitted by thrips, aphids, leafhoppers, and whiteflies. Generally, it is transmitted by thrips tabaci. The metalaxyl is a specific fungicide that is recommended against. So for all the in the previous classes or in the previous presentations, I think I have already discussed in the earlier published videos also. If you see the disease of crops also, I have clearly told that metalaxyl is the specific fungicide that is especially recommended against oomycetes group of fungicides. So for example, if you see Peronosporales, Erysipelas, Moniliales, Mucorales actually Moniliales, Erysipelas that not comes under this Erysipelas powdery mildew genera. This Mucorales actually this Rhizophus mucor, so Jegomagata group of Moniliales. These are all not at all oomycetes group of fungi. Only Peronosporales, for example, all kinds of downy mildews that comes under this. So this all downy mildews, not only downy mildews, for all oomycetes group of fungi, metalaxyl is a specific fungicide. So the answer is Peronosporales. So the next one in 1927, who discovered the function of spermatia and the rolla in dikaryotation? Craig, Bowden, Trumlachar, Casey Mehta. So answer is Craig. In 1927, Craig was first to discover the sexual stage in wheat stem rust, that is otherwise called as spermatial stage. So it was first discovered by a Craig. F.C. Bowden, he is actually a virologist. He called virus is a nucleoprotein, which means it is a combination of nucleic acid and protein. Thirmulachar means we know very well antifungal antibiotic. The first of antifungal antibiotic Ariofungin was discovered by Thirmulachar only. And K.C. Mehta, father of Indian Rus. Solen suit of cocoa or cocoa solen suit is actually transmitted by Pentalora nigronervosa, Pseudococcus nijalensis, Nephrotitis synthesipes, Apis crassiflora. So here actually this cocoa solen suit is actually transmitted by mealybug. So here the scientific name of mealybug is Pseudococcus nijalensis. So the answer is B. The next one is Agriculture Scientist Recruitment Board was established in the year. So November 1, 1973, 5, 1973, December 1, 1973, January 1, 1974. I think it's a November 1, 1973. Agriculture Scientist Recruitment Board was established in the year November 1, 1973. So which of the following is a sexual resting spore? So iridospore, teleospore, chlamydospore, and zoospore. So chlamydospores and zoospores are not at all sexual spore. So if you see iridospore and teleospore, iridospore is called as a repeating spore, whereas the teleospore is a resting spore. I think in the previous slide itself I have mentioned about this rust and smut. Iridospore is called as a repeating spore, summer spore, teleospore is called as a resting spore, winter spore. I think I have given there itself. So the next one is asexual resting spore. Ascospore, oospore, basidiospore, chlamydospore. Here ascospore, oospore and basidiospore are the sexual spore, and there is only one spore, that is a chlamydospore, which is a asexual resting spore. The next one is monograph of the fungal Cercospora was written by Cercospora. Actually C. Booth is related to Fusarium. Cercospora is C.D. Chupp. C.D. Chupp is the person who wrote the genera of this Cercospora, but C. Booth is actually related to Fusarium. Next one, QTL stands for quantitative triad loci, qualitative triad loci, quantitative trial loci, quantitative trials locus. So the answer for QTL is quantitative triad loci. So the answer is A. This quantitative triad loci was first given by Gelderman in 1975. Next one, who discovered PCR? So Goodwin and Bantari, Paul Herberts, Adam and Clark, and Carey Mullis. So the PCR was first invented by Carey Mullis in 1983. For the discovery of PCR, Carey Mullis got a Nobel Prize. So Adam and Clark was renowned for the discovery of ELISA. Paul Hebert DNA barcoding. This Goodwin and Bunter, I think it's these two scientists are actually related to some kind of technique, but actually forgot. But this PCR is definitely Carey Mullis, Adam and Clark, ELISA, Paul Hebert DNA barcoding. How many number of wells are present in ELISA? First of all, we must know what is ELISA, enzyme linked immunosorbent assay. So in enzyme linked immunosorbent assay, a plate was there. this kind of plate will be there. So how many number of wells are present, they are asking. 94, 95, 96, 97. Generally, if you see ELISA plate generally contains 96 wells. There are plenty of plates which is made up of 24 wells, 48 wells, 96 wells, 384 wells, and other plates are like 5300 wells also be available. But the most commonly used ELISA plate contains 96 wells. Generally the ELISA plate contains 96 wells. 12 into 18. The next one is which one is called as candle snuff fungi? Xylaria hypoxylon, Xylaria polymorpha, Serpula lacrimens, Claviceps fusiformis. So which of the following fungi is generally called as candle snuff fungi? Xylaria hypoxylon is generally called as candle snuff fungi. Xylaria polymorpha is generally known as dead man's finger. Serpula lacrimens is a house fungus, like some kind of infections it causes. I think it's household fungus, some kind, actually I forgot. Claviceps fusiformis, which cause generally ergot diseases. And Oomycetes cell wall is made up of, generally fungal cell wall is made up of chitin, whereas Oomycetes cell wall is made up of cellulose plus hydroxyproline. A little amount of hydroxyproline is also present. So chitin means fungi, glucans, chitin and little amount of glucans are generally present. But when Oomycota cell wall, it is actually made up of cellulose plus a little amount of hydroxyproline. Chitosan chitin, I think yeast cell wall is made up of chitosan chitin. The next one is the name a fungi from Oomycota whose cell wall is made up of chitin instead of cellulose. Sorry, the question was actually blocked by this picture. Anyway, I will tell you the question. Name the fungi from Oomycota those whose cell wall is made up of chitin instead of cellulose. Acaila, Aphadogyla, Saprolegnia, and all the above. So the answer is all the above. There are only couple of fungi from Oomycota. Even though they comes under Oomycota group of fungi, their cell wall is made up of chitin. Example: Acaila, Aphadogyla, Saprolegnia, and one more fungi is also there, Leptometus. So these four fungi actually, even though they comes under Oomycota, their cell wall is made up of chitin. Five kingdom classification was given by Carl Woese, Whittaker, Linnaeus, and Ainsworth. The answer is actually Whittaker 1969. On the right side, I have given one picture where this entire classification was given here. Actually, two kingdom classification was given by Linnaeus. He is Linnaeus. So the two kingdom classification is Plantae and Animalia. So three kingdom classification was Ernest Haeckel. The three kingdom is Protista, Plantae, and Animalia. So about Copperland, four kingdom classification was given by Monera, Protista, Plantae, and Animalia. Five kingdom classification is R.H. Whittaker, Monera, Protista, Fungi, Plantae, and Animalia. So Carl Woese six kingdom classification was given Eubacteria, Archaebacteria, Protista, Fungi, Plantae, and Animalia. Chiton, two empire kingdom that was actually given Eumycota and Eukaryota and Prokaryota. Carl Woese three domain classification, bacteria, archaea, and eukarya. So here the five kingdom classification, that is the question that was given by Whittaker. So if you guys want a detailed explanation about this classification and all those things, I have clearly given in my mycology section videos. You just go to mycology section, that too in classification of fungi. I have given very detailed and classification from the starting from CH Poisson to the almost advanced classification, that is Kirketal classification. So the next one is vector for bacterial wilt of corn, Chitonema pulcharia, corn flea beetle, glassy winged sharpshooter, both A and B. So the answer is both A and B, because it is transmitted by corn flea beetle. The scientific name of corn flea beetle is Chitonema pulcharia. So the answer is both A and B. So the next one is expand ISEM. ISEM means immunosorbent electron microscopy. I think the answer is A. The next one is which one is the first plant pathogenic fungus to be completely genome sequenced? Magnum porte grigia, Sclerotian ralphsi, Alternaria solani, Fusarium solani. So for the answer for the next four questions, I have given here in a small table. So the first fungi to be completely genome sequenced is Magnoporthe grigia, sequenced by D. Natal in 2005. I think I have also given the genome size 40 MB. So bacteria, first bacteria to be completely genome sequenced is Xylella fastidiosa, which is a Pierce disease of grapevine, which is AJG Simpson 2000. The size is 2.68 MB. The next one, first DNA virus to be completely genome sequenced, cauliflower mosaic virus, Frank et al. 1980. First RNA virus to be completely genome sequenced is tobacco mosaic virus, that was sequenced by Golet 1982, 6.3 to 6.5 kb. First viride to be completely genome sequenced is potato spindle tubed viride, that was sequenced by Gross et al. 78, Dixon 79. The next one, yeast Saccharomyces cerevisiae 1996, 12.1 MB, and the nematode is Chinorhabdis elegans, that was sequenced by Andrew Fire and Craig Mello 1998, 100 MB. Now the question we will see, what is the first plant pathogenic fungi to be completely genome sequenced? Pyricularia varizae, the teleomorphic is Magnoporthe grigia, that was sequenced by Dean et al. Next one, rice blast generally. Which one is the first plant pathogenic bacteria to be completely genome sequenced? Plant pathogenic bacteria means Xylella fastidiosa. First DNA virus to be completely genome sequenced, cucumber mosaic virus. First RNA virus to be completely genome sequenced is tobacco mosaic virus. So for a easy understanding, I have placed a picture here on the right side top, so that will be, hope it will be helpful. The next one is Rabigale festival is celebrated on, generally the Rabigale festival is celebrated on April 25th of every year in the Roman calendar. So this Robigolia festival was actually initiated by Numa Pimpilus, a king around 700 BC. It is especially meant for rust god. The rust god name is Robigus. Next one, who proved that the presence of murine in the bacterial cell? Vidal, E.F. Smith, Robert Koch and Graham. So the first person who proved that bacterial cell contains murine, that is Vidal. So size of TA plasmid 200 kb. So Berg's manual of determinative bacteriology was published in there. There are two kinds of manuals are here related to bacteria, naming, classification, everything. So Berg's manual of determinative bacteriology and Berg's manual of systemic bacteriology. Generally, the Berg's manual of determinative bacteriology was published in the year 1923. When it comes to systemic bacteriology, 1984. So later editions also will come, volume 1, volume 2 edition or something like that will all come. It also came, but generally the first was published determinative bacteriology means 1923, systemic bacteriology means 1984. Then phylum Plasmodioporales belonging to the kingdom. Generally Plasmodioporales is not a true fungi, it's a fungi-like organism. Generally if you see in the classification also, it was actually classified under protozoa. Okay, chromista means oomycota group of fungi, all those will come under. So the answer is protozoa. The next one, average speed of a bacteria per second. I think you might have studied this question in various other books. Generally the speed of bacteria is 30 to 50 micrometer per second. So the teleomorphic stage of Cercospora, here Cochiliobolus, Pleospora, Mycospirilla, Glomerella. So Glomerella means a Coltrotrachum, I think. So Cochiliobolus, that is bipolaris, and Pleospora I have forgot, but when it comes to Cercospora, it is Mycospirilla. Then what is the most sensitive technique used for the detection of plant pathogens? PCR, RFLP, RAPD and ELISA. I think ELISA, about ELISA we have already discussed. ELISA is the most sensitive technique used for the detection of plant pathogens or plant viruses. ELISA means enzyme linked immunosorbent assay, that was first discovered by Adam and Clark in 1977. So the ELISA plate generally consists of 96 cells. Next one is what is curing? So elimination of DNA, elimination of plasmid, elimination of cell wall, elimination of mesosomes. Generally elimination of plasmid is known as curing in when it comes to this pathology, but generally the process of curing is also used in tobacco industries just to get quality produce or something like that, maybe heating or something. I don't know exactly what it is, but when it comes to pathology, it is just elimination of plasmid is known as curing. So which pathogen entry through trichomes? Bacterial canker of tomato, bacterial leaf streak, black rot of cabbage, potato scab. So this black rot of cabbage can enter into the plant cells through hydrodes. So bacterial canker of tomato then enters the plant cells through trichomes. Next one, diamond shaped symptoms are produced by Pyroclaria varizae, Alternaria solani, Magnoporthe grigia, A and B. So this Pyroclaria varizae and Magnoporthe grigia both are same, so the answer is A and B, because this Alternaria solani, the specific symptoms is generally all Alternaria plant pathogens produces concentric rings. This diamond shaped spots are produced by blast pathogen. Generally rice blast is caused by general rice blast is caused by Pyroclaria varizae. The other teleomorphic stage Magnoporthe grigia. Polyatic diseases are produces. So here in epidemiology, if you see monocyclic diseases, polycyclic or polyatic. Okay, so here the polyatic diseases are generally produces a bimodal curve. Monocyclic generally produces a saturation curve. Polycyclic sigmoid curve or S-shaped curve. Polyatic bimodal curve. So which of the following is a magnesium deficiency? Pala beta sugarcane, mur spot of pea, grey speck of oat. It seems to be that all the above diseases are actually caused by magnesium deficiency. Pala blight of sugarcane, mur spot of pea, grey speck of oats, all. So DIP Act was passed in the year 1912, 41, 14, 46. So actually Federal Quarantine Act was the first quarantine act that was passed by America in 1912. So later the same act in India was enforced with another name called DIP, Destructive Insect and Pest Act, in the year 1914. In 1946 is also an important year where DPPQ is started, Directorate of Plant Protection, Quarantine and Storage, located at Faridabad, Haryana. Chestnut compound was developed by, so chestnut compound was actually developed by Beverly. Merada developed Bodomixture, Mason developed chestnut compound Beverly, Mason Bodomixture, Burgundy mixture, I think Mason 1887, Millard 1880 to 85, Bodomixture, Beverly 1921, chestnut compound 2:1, sorry 2:11. The next one, which one is a symplastic movement fungicide? Metaloxyl, carbendazim, phosphatal aluminum, oxycarboxyn. Actually, symplastic means both acropetal and basipetal. The first preference was given to phosphatal aluminum. If phosphatal aluminum was not there in the option, then we can go with the metaloxyl. Here the answer is phosphatal aluminum, and actually both the answers will come metaloxyl and phosphatal, but first preference will go to phosphatal aluminum. So name the book, the book written by Y.L. Nene. So Y.L. Nene, he wrote a very famous book called Fungicides in Plant Disease Control. First plant defense activator ActiGuard CGF 245704, both A and B. Plant guard, actually ActiGuard and CGF 245704 both are same only, so the answer is both A and B. Expand lamp, lap mediated isothermal amplification, loop mediated isothermal amplification, loop amplified isothermal purification, loop mediated isothermal purification. So the answer is loop mediated isothermal amplification. It is a technique that was developed by Notomi in 2000. It is nothing but it's like a PCR only. In PCR 3 steps will be there, in lamp only 1 step and only 1 temperature will be there. That is the only thing. It is also DNA amplified technique, that's it. So the first book that is completely devoted to fungi. Okay, Theotrum fungorum, the disease of cultivated crops under their control, Nova plantarum genera, Synopsis methodica fungorum. Actually the first book that is completely devoted to fungi is Theotrum fungorum, that was written by Van Streebuck, I think 1675. This is actually the first book that is completely devoted to only about fungi. So before also there are a couple of books, so all the books maybe it's like a mingling of all the concepts, like some kind of books like involved about plants and mentioned about plants, some classification, some kind of other things will be there, but this is the first recorded book or reported book to be completely devoted to the fungi, Theotrum fungorum. So here this book is the disease of cultivated crops under their control. This is the first plant pathology book, I would say first book that is completely devoted to plant pathology, especially diseases. That was written by J.G. Kuhn in 1858. This book is considered as the first book in plant pathology. So the next one is the Nova plantarum genera, that was written by P.A. Michel 1729. Synopsis methodica fungorum, I think C.H. Persoon 1801. So sensitivity of ELISA 1 to 10 picogram per ml, 1 to 10 nanogram per ml, 1 to 10 some things will be there, but the answer is 1 to 10 nanogram per ml. This is the most sensitive technique that is developed. So the next one is measuring the size of the minute objects in a microscope is known as micrometry, pathometry, turbidometry and micrometry. So micrometry is the general technique which is used to measure the small objects in the microscope. So which disease is called as Mission Impossible in quarantine? Rice blast, kernel bunt of wheat stem rust, all the above. I think this kernel bunt of wheat is generally known as Mission Impossible disease in quarantine, because it is very difficult to control. That's why it is known as Mission Impossible disease. Blotter test is used for seed health testing, seed vigor, seed purity, seed index. Generally we know this blotter test is generally recommended for the testing of seed health. So the answer is A. Hypertrophy means abnormal enlargement of cell, increase in number of cells, decrease in number of cells, decrease in cell divisions. Hypertrophy and hyperplasia, there are two terminologies here there. Hypertrophy means abnormal enlargement of cells. Hyperplasia means increase in number of cells or cell divisions. So here hypertrophy means abnormal enlargement of cells on cell. Epidemiology of wheat stem rest life cycle was made by K.C. Mehta, Nagarajan, Mehrotra and Munkur. So generally this epidemiology of life cycle and all those things was first started in India by K.C. Mehta. That's why he is known as father of Indian rest. So Nagarajan, he made breakthrough contributions on wheat stem Puccinia pathway and all those things. And B.B. Munkur, we know very well, he is the man who started IPS, Indian Pathological Society. Effective fungicide for the control of rice blast. You always remember something, for the rice blast there is a specific fungicide there, tricyclazole. The trade name is beam blastinius. Various kinds of trade names are there, but the specific fungicide recommended for rice blast is tricyclazole. Cleistothecium with several asci with coiled tips. So coiled tips itself indicates it's uncinella coiled tips. This regarding this Cleistothecium and the kind of appendages with one ascus and many ascus in I have already discussed in detail about this types of appendages in model question paper number 14. I think if you might not have if you have not seen that one, you better to go and take a look into that. So there I have mentioned other information regarding this Cleistothecium types of appendages also. But here the answer is if it is a coiled tips, it is uncinella. Next one is the genome of Gemini virus, single stranded DNA, single stranded RNA, double stranded DNA, double stranded RNA. Generally, this Gemini means a twin particle. So this the genome of Gemini viruses contains single stranded DNA. Next one, International Mycology Institute located at Kew, England. It was started at around, I don't know, maybe 1920. The first president of this International Mycological Institute is E.J. Butler, father of Indian plant pathology. So brain flotation method is used to identify wheat loose smut, wheat tundu disease, grain smut of Java, rice blast. I think wheat tundu disease. For the identification of wheat tundu disease, we can use this technique called brain flotation. Just the infected grains normally used to float on the solution. That's it. So genome of most of the plant viruses, RNA, DNA plus RNA, none of the above. So majority of the plant viruses contains RNA as a genetic material, that to positive single stranded RNA. Banana bunch type is transmitted by Escherichia coli, Aphids caseora, Mysis persicki and Pentaloria nigronervosa. Generally, this banana bunch type virus is transmitted by black aphid, Pentaloria nigronervosa. There is one more disease is called Cuttay disease of cardamom. Banana bunch type virus, Cuttay disease of cardamom. These two diseases are actually transmitted by black aphid, Pentaloria nigronervosa. Of course, there are a couple of viruses also that can transmit through this black aphid, but these two are important. Banana bunch top, Cuttay disease of cardamom, both are transmitted by black aphid, Pentaloria nigronervosa. And what are all the spores produced by wheat stem rust fungus on barberry? Again in the question paper number, model question paper number 13 or maybe 14, I have discussed about the various kinds of spores that is produced by this rust fungi. Generally, two kinds of hosts will be there, primary host and secondary host. The primary host is a wheat. In wheat, teleospore and iridospores are produced upper surface. If you see the secondary host is barberry. On the upper surface of barberry, pycniospores are produced. On the lower surface, Asium is produced. So here the answer is both A and B, pycnium and Asium. So fungicide recommended for grapes powdery mildew. So grapes downy mildew we can recommend bodomycin. For the grapes powdery mildew we can recommend a fungicide called calixin. I think it's a triadimifon. The trade name of chemical name is triadimifon. Triadomorph is Baileton, triadimifon calixin, I think. So the next one is a bacteriophage. Phage means eater. Bacteriophage means bacterial eater. A virus that infects bacteria is known as bacteriophages. It was first discovered by Twart and Herrelli. So the answer is A, virus that infect bacteria. Most phytoplasma are transmitted by aphids, leafhoppers, mealybugs, and mites. Most phytoplasmal and spiroplasmal diseases are transmitted by leafhoppers only. Majority of the phytoplasmal and spiroplasmal diseases are transmitted by leafhoppers. Sexual spores produced by the downy mildew fungi. All downy mildew fungi produce sexual spores as oospores. Alternate host for Sesamum phyllodes, Sclerocarpus africanus, Brassica campestris, Osimum canum, and all the above. Normally, the Sesamum phyllodes, Orochius albicinctus, leafhopper, and I think they have mentioned the other things rather than that of this Orochius albicinctus. Probably the answer is Sclerocarpus africanus, I think. The next one is a perfect stage of Aspergillus nodulans. Aspergillus nodulans perfect stage, Emersella. Then next one, father of Indian rust, I think we have already discussed, K.C. Mehta, tyndallization temperature. So generally I will tell you, this technique was actually developed by John Tyndall for the killing of endospores. So the technique is known as tyndallization. So these endospores are highly resistant to it, that can survive up to 100 degrees centigrade. There is a special technique for the killing of these endospores is known as tyndallization. It was developed by John Tyndall. Robert Koch, John Tyndall are individually discovered this tyndallization technique, but the credit goes to John Tyndall because he is the one who first discovered it. So the tyndallization temperature is 100 degrees Celsius for 20 minutes for 3 consecutive days. Next one, kidney shaped radiospore and turnip teleospore is the characteristics genus of Hemilia. Hemilia produced kidney shaped radiospore, kidney shaped radiospore and turnip shaped teleospore. Puccinia means double celled, Euromyces single celled ospore, Melonospora I think crust like teleospore. So Bakane disease is due to auxins, gibberellins, cytokinins and ethylene. So gibberellins are actually associated with Bakane. So it was first discovered by Kursova. Kursova was the first one who discovered this gibberellins. This Bakane disease of rice is actually associated with the gibberellins. That's why it is otherwise called as foolish seedling disease, because the infected seedlings are actually much more taller than that of normal seedlings because of production of this gibberellin. Generally, it is a growth hormone. Bimodal transmission can be observed in Gemini virus, cauliflower mosaic virus, bromovirus, tobamovirus. So bimodal means cauliflower mosaic virus actually transmitted through aphids and as well as sap transmission. Alternate host for Puccinia graminis tritici, Barbaris vulgaris, Oxalia corniculata, Mahonia and tomato. We know very well Barbaris vulgaris, Barbary. Next one, tomato leaf curl virus is transmitted by Thrips tabaci, Bimsia tabaci, Mysis persicale, Pendula nervosa, tomato leaf curl virus. So I think if you see, majority of the leaf curls is generally this Bimsia tabaci only. I think as far as my I think probably. But the answer for this tomato leaf curl is Brimsia tabaci. Quarantine regulations for Coliroga of areca nut is present in. So this Coliroga areca nut was first discovered in Karnataka. Later is also discovered in Tamil Nadu as well as Kerala also. But generally it is first identified in Karnataka. So this is the quarantine regulation of areca nut present in Karnataka. Next one, indicator plant for citrus Tristeza virus. So on the right side I have mentioned the various indicator plants. So when it comes to this answer, the indicator plants for citrus tristeza virus is Claxi lime. Solin suit was actually transmitted by, I think this question was previously came, mealybug, aphids, black aphid and leafhopper. Generally it is transmitted by mealybug, Pseudococcus nigerlensis. So the next one is, what is the phenolic compound produced in plants in response to the fungal infection? Glycoproteins, proteins, lipids and phytolauxins. I think we know very well it is a phytolauxins. So these are all kinds of, phyto means plant, alexin means protecting substances which is actually produced by the plant against invading pathogens and abiotic stress also. And citrus tristeza can be effectively managed by budding, grafting, cross production resistant varieties. So citrus tristeza virus is effectively managed by a technique called cross protection. So this technique was actually developed by McKinney against tobacco mosaic virus. But this technique was very effective against this citrus tristeza virus, so that nowadays we are exploiting against the citrus virus, but this cross protection technique was actually developed against tobacco mosaic virus. Simple interest diseases are produces a curve. Simple interest disease means monocyclic. Compound interest disease means polycyclic diseases. Here the simple interest diseases produces saturation curve. So bimodal means polyetic. Sigmoid curve or S-shaped curve means polycyclic disease or compound interest disease. So Puccinia produces Aeschiospores. I think in previous also we have discussed Aeschiospores and Pycniospores. These two spores are produced on secondary host, that is a Barbary. Primary host, wheat, teleospores and iridospores are produced. The answer is Barbary. Flagella type of Irvinia. Monotrichus, amphitrichus, peritrichus, cephalotrichus. So the flagella type of Irvinia is peritrichus flagella. Monotrichus means single flagella. Atrichus means no flagella. Example is Xylella. Everything was given here. Just to take a look into. Atrichus means no flagella, xylella. Monotrichus means single trichus, xanthomonas, amphitrichus, peritrichus, lobotrichus, and all those things was given here. Just take a look into. So the next one is hartignates are found in ectomycorrhiza, endomycorrhiza, ectoendomycorrhiza, all. So generally these hartignates are found in ectomycorrhiza. This is also very important question that was repeatedly asked before. Single ascus with mycelial appendages can be observed in Erisife, Levellula, Podospora, and Sparotheca. Generally, this single ascus with mycelial appendages are found in Sparotheca. Blotter paper test used for the detection of. So, blotter paper test generally used for the detection of seed-borne fungi. Sexual spores produced by Deuteromycota. So Deuteromycota produce no sexual spores. Actually, this Deuteromycota is called as dustbin fungi or fungi imperfecti because having no sexual stage. So that's now it is named and sorry, now it is classified under Ascomycota. Generally, Deuteromycota means no sexual spores. Wheat tundu is caused by Angana triticale, Corynebacterium triticale, Ratibacter triticale, all the above. Generally, wheat tundu disease, this Angana triticale is associated with another bacterium called Corynebacterium triticale. Now it is called as Ratibacter triticale. We can also call as Clavibacter triticale. Okay. Now the answer is all the above. Now this wheat tundu disease, otherwise called as yellow slime rot disease. This is actually these two diseases are actually association of bacteria along with the nematode Angana triticale. This is associated with a bacterium called Corynebacterium triticale, otherwise called as that is a Ratibacter triticale. We can also call as Clavibacter triticale. So the answer is all the above. Non-motile spore. Zoospore is motile. Aplanospore is non-motile. Zygospore is also yes motile. Planospore is also motile. Planospore is also fungi. Okay, so the answer is aplanospore. Aplanospore is non-motile. The next one is a foot rot of pepper is caused by Pedoptera infestans, Pedoptera palmivora, Pedoptera parasitica, Pedoptera capsicum. Generally, foot rot of pepper is caused by Pedoptera capsicum. Rice blast was first discovered in India, China, Japan, Finland. So the first discovered in China in 1637. So in 1637, the first one is reported is China. They named some other name, actually I forgot, but the China was the first country who reported this rice blast disease. 1637. Trade name of oxycarboxyn. And I forgot to tell you one more thing. This the first rice blast was first discovered in the country China. That was the net question in 2023. If you see the net question paper 2023 that was published in our YouTube channel also. So this rice blast was first discovered in China 1637. That is also 2023 net question net asked question. Trade name of oxycarboxyn. Oxycarboxyn plant wax. Carboxyn Vetavax, Redomil, Metavaxel, Redomil Gold, generally metalaxyl plus carbendazim combination. So the Bevestin mancozib. Then repeating spore, iridospores are iridospores, achiospore, basidospores, and teleospores. Iridospores are repeating spores, teleospores are resting spores. Year of lead blight of potato, everyone knows, 1845. Pathogen derived from protein coat, divide of protein coat. Divide means no protein coat are present, that is divide of protein coat, viride. So virus contains protein coat and nucleic acid. Virus is divide of protein coat. So Pytoplasma also contains no cell wall. Instead of cell wall, it contains a triploblastic membrane. So bacteria obviously contains cell wall, that is peptidoglycan, smallest viride in the world. Potato spindle tuber viride, coconut kadang viride, chrysanthemum chlorotic mottle viride, citrus exocartus viride. And I think I have already told you coconut kadang is the smallest viride which contains 246 nucleotides, and the largest viride is chrysanthemum chlorotic stunt viride which contains 398 to 401 nucleotides. So citrus exocartus is probably 371. Potato spindle tuber is probably 357, I think, nucleotides. So these are the answers for the questions. And we have for easy understanding, we have placed first reports of all the organisms. For fungi, first scientist who report this fungi is Anton de Berry, in the sense which causes generally plant disease. Before also it was reported, but generally when the new era of plant pathology was started, fungi, Anton de Berry, lead blight of potato, Phytophthora infestans. Bacteria, TJ Berry 1878, fire blight of apple and pear, Irvinia amylovora. In India, bangle blight of potato was the first disease that was reported by scientist TJ Butler. The virus, first virus reported is MJ Bizernick. tobacco mosaic virus, which causes tobacco mosaic disease. In India, the first reported virus is sugarcane mosaic virus that was reported by J.F. Daster. He is the second president of Indian Phytopathology Society and also renowned for the Phytophthora diseases in castor, seedling blight of castor, I think Phytophthora parasitica. So viroid, Diner and Raymer, potato spindle tuber viroid, which causes potato spindle tuber disease. In India, it is a tomato bunch top that belonging to the citrus hexocortosis group. Virucide, Randless, valvete motile virus. And next one is phytoplasmal disease that was reported by Doyle et al. First phytoplasmal disease in 1967. Mulberry dwarf. It was transmitted by leaf hopper, Hishimonas piscitis. In India, it was the sandal spike. That was the first phytoplasmal disease that was reported by Verma 1969. Spiroplasma, Davis et al. Constant. That was actually transmitted by leaf hopper Dalbus medis. Prions, Stanley prisoner. Kuru disease or scrapidaceous sheeps. Satellite virus Casanis. Tobacco satellite necrosis virus. Mycoviruses Hollings. So Lala France disease of mushrooms, otherwise called as X disease or water stripe disease. Cyanophases, Saffroman and Morris, LPP. So bacteriophages were Twart and Herilli. So these are all the some couple of first reports. So I think we might expect a couple of questions from this. So hope it will be useful. So for much more information and a detailed description about various kinds of information was mentioned in my book, A Vision into Plant Pathology, complete student version. So for further doubts and clarifications or guidance, you guys can reach us at www.geekyresearcher.com. Stay geeky and stay tuned. We are Team Geeky Researchers.